the argon group, what you can be telling us about something called organovanadium chemistry of oxides.
Cool. Great. Thanks for the introduction. So today we're going to talk about some of the computational work that we're doing in the argon group, and the catalysis group, mainly about talking about the support organovanadium catalyst and how we can investigate this computation mechanistically and through XANES simulations. And so we've heard about this in a couple of talks earlier today, and the previous one about the single atom catalyst, and the idea of the surface organometallic chemistry and catalysis in general. So this morning's sessions, I'll give a great overview of what surface organometallic chemistry is. So as a quick recap, it's basically the idea of how we can have the stability of heterogeneous catalysis with the high selectivity of homogeneous catalysis, and having essentially these organometallic complexes that can graft onto the surface. Now, some of the features that we're kind of interested in from a characterization standpoint is the idea of the X-ray absorption spectroscopy, which does measure an aggregate of sites, but if we try to go through XANES or XAVs, so in particular for XAVs, it's hard to identify things like metal hydrides, which we know are essential in various catalytic processes. So we don't want to make sure that we can figure out how we can try to figure these out from a computational point, but also the idea of second shell coordination in terms of some of these metal oxide or metal metal interactions that can occur from the nature of having single atom catalysts on a metallic surface. So the catalysts of interest for us are these amorphous silica structures. Now, part of the idea and the complexity of it is that we're dealing with an amorphous structure, which adds an extra layer of complexity in terms of a modeling standpoint, where just from an amorphous silica surface, we have at least 4 different types of bonding configurations through monopodal, bipodal, or even tripodal bonding configurations, which leads to a distribution of potential bond distances between 2 or 3 neighboring oxygen atoms that all graft to a single metal atom. And so from that point, and then also through X-ray spectroscopic methods, you kind of target an aggregate of sites, but these aren't necessarily the most catalytically active. So what can we do from a computational point and needing these additional models to show the effect of site heterogeneity and how this affects the catalysis? And so our group previously synthesized and characterized a hydrogenation active supported catalyst through an amorphous silica, vanadium-3, with an acetylene and a THF ligands down to the silica surface. So their previous study on the mechanistic and kinetic profiles showed that this catalyst has behavior for hydrogenation, and when they treated this pre-catalyst with hydrogen in the XANES spectrum, they noticed an increase and a broadening of the pre-HP shown in red versus their catalyst as treated. And so from a computational point, as I mentioned earlier, we have these metal hydrides that aren't necessarily detectable by XAVs, or at least difficult to. So from a computational standpoint, what can we figure out from a hydrogen treatment? What can we gain from these computations? To solve this puzzle, we're going to do this into 2 parts. One talking about the mechanism, and a second part talking about XANES analysis through these methods. And so the quick overview is that we use basically a cage model. So this is coaxium cages to model in amorphous silica. And the idea is that these cage models can represent the local interactions of a silica surface, but allows us the tunability and computational feasibility that molecular programs allow us, and electron circuit programs allow us to have, versus some of the standard periodic models that we see for heterogeneous catalysis. So to do this, we can look at time-dependent DOT for excited state or even XANES calculations, and so methods like these are more practical for molecular cages and periodic models. This is why we're going down a DFT, time-dependent route. But then we can also start looking at these heterogeneity aspects. So here we're going to focus on 2 degrees, or 2 disordered parameters, one being the vanadium oxygen from a donor siloxane. As we'll show, this is kind of more important in the catalytic process and part of the proposed mechanism. But then we can also look at the graphene site heterogeneity through this particular oxygen-oxygen distance caused by ring strain on the surface. And so we can see how these could potentially be tuned and how that would affect the catalysis and other processes moving forward. So the first part is we're talking about the mechanism that we have. So we started with our pre-catalyst of vanadium-3 with the THF and the acetylene ligand. It follows a sigma bond metathesis main step to form the hydride, and then through, in this particular case, it was styrene hydrogenation. So we can see how through the various steps, we can see how we have 3 potential active sites. One being the bare site with the full silicon model. We have a THF that's laid bottom on the surface, so it can interact with it, whether as part of THF or not. I'm not sure why the reaction arrows not pop up, but then with the styrene that can also bind to it. So we have 3 potential active sites, and one of the main observations we can get from this particular reaction profile from a DFT standpoint is the differences between the observed and the intrinsic barriers, which do show about 20 kcal per mole energetic difference, and there's a little bit more when we talk about separating these by a vanadium oxygen bond distance as well, and how that goes through the reaction profile. So following the active site formation, we can now go into the proper styrene hydrogenation reaction profiles of all 3 potential active sites. And so here we're modeling site heterogeneity based off the bond elongation of the various active sites, being A, B, and C, and using 3 different vanadium oxygen as well as a full geometry relaxation. This allows us to get a minimum on that potential energy surface, but what we can see from that is that through these relaxed structures, it actually generates the highest barriers. So while it's pretty standard to use a fully relaxed geometry for computational work, having these higher barriers showed that having a fully relaxed structure resulted in these metastable states, which makes catalysis a little impractical compared to increasing the bond distance, which decreases the stability and increases that reaction energy. Decreases the barrier. Then you can see that at longer distances, generate a lower energy profile. And then through this particular reaction mechanism, this was consistent with the kinetic analysis. It has the reaction in the same order for the hydrogenalysis of the various catalysts. So from that, we can say that from just having the lowest barriers as well, that this bare site is probably the most active. And so we can go into further investigations to see what that contains. So in a collaboration with Professor Rick Skoji at the University of Colorado Boulder, they looked at using kinetic Monte Carlo simulations, which the idea is using a Monte Carlo sampling of a catalytic reaction profile, using the various structures and DFT parameters to generate these kinetic profiles, and each generated at various distances. And so at the top we have the experimental kinetics that were performed in the initial study, and in the bottom 3 graphs here we have the computationally generated kinetic profiles. And so the qualitative shape is mostly the same. Reaction order is a little bit off, but that's mainly due to the fact that DFT is not a quantitatively accurate measurement in this particular case. Metals is a little bit complicated with that too. But the main idea is that we can get this qualitative shape in the curves and mimic the order of the experiments. Another part of it was that we had to use a linear combination of the various distances in order to generate qualitatively consistent kinetic profiles. And we do have follow-up studies as well, kind of going more in depth on the kinetic aspect of the modeling. So this kind of showed us that having this combination of kinetic Monte Carlo profiles is needed to qualitatively describe these experimental kinetic profiles as well. And so now we kind of idea of the mechanism, and now that I was involved in that, now we kind of shift our focus to the XANES portion of this. And so focusing on that pre-edge region that we talked about earlier, where we had the increase and the broadening of the pre-edge peak, we can see how we can try to calibrate this with various vanadium complexes, since the idea is doing vanadium K-edge XANES on a silica surface. So we built a data set of various vanadium complexes with vanadium oxygen, nitrogen, organometallics, vanadium chlorine, selenium, various ligands. About half of these came from Dan Mignola's group at the University of Pennsylvania. And so we characterized those at the APS facility at Argonne, as well as grab some data from literature. And through this calibration technique that we can see, one, we have qualitative trends in the ligands and the oxidation states, but we also have a calibration method that is both quantitatively accurate, generating a mean absolute error around 0.4 EV in terms of predictions, as well as a qualitative consistent, through which you can see in these violin plots, which through the functional that we selected, we did a very rigorous test on computational parameters. But through these qualitatively consistent violin plots, what we can see is that the distribution of vanadium 5, 4, 3, 2, etc., oxidation states, we can see how that distribution plays out, and through our calculated versions, how those distributions are similar, at least in terms of where they're weighted as well. But we can also see that there's a trend in terms of vanadium nitrides for the vanadium 3 and vanadium 5, having a lower pre-edge energy than the vanadium oxides for the same species as well. So you can get some more intrinsic trends that we can kind of pick out just from and organizing these by ligand classification as well. So now that we have this calibration method, we can look for, going back to that problem of the site heterogeneity and the hydride formation. And so the main gist of it is that we can use this concept of what's called natural transition orbitals, and we can separate out individual contributions by ligand type on a particular complex. So from here we can see that the metal hydride is more sensitive to bond elongation of the oxygen-oxygen disorder parameter that we have, and that's more sensitive to site heterogeneity than the pre-catalyst, as we can see by a rough estimation of the broadness being 0.62 based off bond elongation versus about 1 EV shift. So the main takeaway here is that this broadening can be due to site heterogeneity, and obviously this one disorder parameter is not enough to account for all of it, but it is a portion of it. So we can see, at least in terms of the modeling standpoint, how these kind of rudimentary models, at least at the starting point, to figure out the true disorder that's going on in modeling in aggregator sites through XANES. The second part of our characterization that we were able to obtain through this XANES study was figuring out these second shell coordination effects. This kind of gave us an insight into whether metal clusters or single sites are formed on a catalyst. As a catalyst, we do want to make sure that we can have techniques that can help us identify what's going on for those particular cases. And so part of these vanadium nitride species, these are ones that were synthesized by Dan Mignola's group in particular. But the idea is that since the first shell coordination is all vanadium nitride, in order to properly analyze this, we have to move to the second coordination to figure out what's going on. Now, through the experimental fitting, we're able to see that there's 3 potential features in terms of these pseudovoid peaks that can generate the experimental pre-edge peak through a convolution. Through a computational analysis, we're able to see how each individual ligand stacks up to this and also aligns pretty well with where the experimental fitting takes place. So one being the vanadium nitride ligand, vanadium azide, as well as this TP ligand, this tris-methylcrosylborate ligand operating as a tridentate ligand here. But we can see that the shoulder peak, so we can also see how these subtle features show significant change in the structure, just for the fact that this one transition a little bit lower represents this larger TP ligand and represents this very subtle feature in the XANES spectra. Similarly, so now we have a single vanadium atom, but in terms of this metal-metal interaction that we can kind of gauge as well. So using very similar ligand structure, so the same TP ligand, but in this case there's a bridging vanadium 3, 4 nitrogen core in the center. This generates 2 unique environments that can be analyzed. Now through experimental characterization, it's not going to account for each of those individually. Computationally, we can go through these individually, create a linear combination, and that's what we get through this plot here. And so through this experimental fitting, again, in the very light gray, we can see that it's a linear combination of two pseudo-void peaks, but if we separate these out by this outer vanadium and the inner vanadium atom, so through the outer two, which are the same chemical environment, we do see that this slight subtle feature here, it actually does correlate to the same TP ligand that we saw in the monomer species. So just expanding through monomer and trimer, we now see how these are, that same interaction contributes to the same subtle features as well, but then having an overlap of this vanadium nitrogen core as well as the vanadium nitrogen interactions, those also contribute to this same overlapping peak that represents the pre-edge as well. And so we can now see how having this combined experimental and computational XANES analysis is really crucial in determining these pre-edge features and how we can interpret that data to further support the characterization of it. So in conclusion, we can kind of see how we have these quantitative models that are needed for site heterogeneity to describe the full scope of the catalytic behavior of various surface and metallic species. And then using computational methods like time-dependent density functional theory and orbital analysis can be used to better understand XANES of transition metal complexes, as well as, again, the surface and metallic chemistry. But then having that mechanistic and XANES cross-combination, as well as combining the experimental and computational aspects of this, allows for that interpretation of subtle features and other parameters that one can get but not the other, but allows for these subtle features to be identified, and that can help us further characterize these species more, things like hydride and second shell coordination. So then the question that we want to try to answer in future studies related to this is what specific features and trends can apply for other computational characterizations of this work, and what can kind of go into potentially data-driven aspects of this as well. And so for acknowledgements, I'd like to thank our 4 PIs of the catalysis program, my supervisor Son, Max, Jeremy, and Dave, our collaborators at the University of Colorado Boulder, Pennsylvania, the APS, and then our calculations that were done locally through an Argon supercomputer, the rest of the group, and everyone for their attention.
Any questions in the audience? So I guess I have one.
Sure.
So you are losing a little something when you simplify the surface into the cluster model. So for example, when you open siloxane fridges, you could lose some information as far as what you're modeling. Is this particular cluster amenable to that sort of siloxane fridge opening that could occur under hydrogenation conditions?
So it's like potentially that could be an issue, which is like, again, it's one of the risks of using a smaller ball to approximate the amorphous silica structure. What we try to do is have like ring strain only focusing on the top monolayer, but we freeze the bottom one to represent kind of the bulk portion of that as well. But definitely different versions of siloxane cage models could contribute to that. And it could be a potential reason that you just mentioned, if that donor siloxane is causing it to lose some of that information. It just shows that we still need more siloxane models on top of that, but then all of that gets more costly in terms of building these models as well. So could it? Absolutely, but also requires different sets of siloxane cage models.
Any other questions? If not, let's thank you for a great talk.